Hello, thank you for joining us today, Dr. Tony Ting and Dr. Isabelle Riviere. We're here to talk about the overcoming the challenges with development and translation of a therapeutic from academia to industry. And I will give you guys each a brief introduction so that the attendees know your backgrounds, and then we can just go into a nice panel discussion about your perspectives. So Dr. Isabelle Riviere received her Ph.D. in cellular and molecular biology from the University of Paris. She initiated her graduate studies at the Institut Curie in Paris and completed her thesis at the Whitehead Institute in Cambridge, Massachusetts. She developed novel retroviral vectors for in vivo long-term expression of transgenes in hematopoietic MFG and SFG, which are widely used in clinical studies worldwide. After completing her postdoctoral work with Dan Littman at NYU, she joined the faculty of Memorial Sloan Kettering Cancer Center in 1999, where she focuses on developing novel strategies for cell therapies and immunotherapies. Her laboratory investigates genetic approaches to enhance various cell types, including T lymphocytes and stem cells, for the treatment of cancer and genetic blood disorders. Over the past 20 years, she has conceived and implemented multiple manufacturing processes for several phase 1 and phase 2 clinical trials under current GMP practices. Her lab currently supports multiple CAR T cell-based clinical trials for the treatment of hematological malignancies and solid tumors. She also investigates immunological functions of CAR T cells in clinical trials in animal models. She is a scientific co-founder of Juno Therapeutics. She has served as a member of the board of directors of the American Society of Gene and Cell Therapy and of the Alliance for Regenerative Medicine. She currently serves on the advisory board of the Center for Commercialization of Cancer Immunotherapy, C3I, in Canada, and the National Science Foundation and Engineering and Research Center for Cell Manufacturing Technologies. Thank you for joining us today, Dr. Riviere. Dr. Ting is the Vice President of Regenerative Medicine and Head of Cardiopulmonary Programs at Athersys. With more than 30 years of experience in cell and stem biology, Dr. Ting has developed expertise in translational clinical studies with adult stem cell therapies and has been responsible for all stages of the development of multistem from the bench to the bedside. Dr. Ting serves on several Regenerative Medicine Society committees, including Chief Commercialization Officer for the International Society for Cell Therapy. From 1995 to 2001, Dr. Ting was a principal investigator and head of the screening for novel inhibitors group at the Institute of Molecular and Cell Biology at the National University of Singapore. Prior to joining IMCB, he was a postdoctoral fellow at Stanford University. Dr. Ting received his Ph.D. in cell biology from Johns Hopkins University. Thank you for joining us, Dr. Ting. So with that, would each of you like to give maybe a moment or two of just your general views on the fields and things like that? And then we can go into kind of just a general panel discussion. So, Dr. Riviere?
Okay, good afternoon. So I guess we are here to discuss the translation of cell therapies from academia to industry. And we know that these partnerships are really important and really are at the essence of the innovation and of the development of new cell and gene therapies. I think that we have made tremendous progress and really breakthroughs over the past 15 years in the field of cell and gene therapies. And I look forward to discussing our experience at Memorial Sloan Kettering Cancer Center in terms of the successes and challenges that we have overcome to bring these cell therapies to fruition to help the treatment of patients with cancer and other diseases.
Very good. Thank you. Dr. Ting?
So I echo Isabelle's comments. I think this is a really exciting time to be in the cell therapy space. From the world of academia, there's novel cell therapy products that are being introduced almost on a weekly basis. And so it's quite exciting to be in the cell therapy space at this moment. Athersys, we in-licensed an adult stem cell technology many years ago from an academic lab. So again, it is kind of the model that a lot of our industry is that you in-license something that an academic lab has developed, and then you have to carry it through to get it through the clinic and ultimately approved and allowed to be given to patients. So this, I think this is actually a very interesting topic for today, and I look forward to participating.
Very good. Thank you. I will start with some general questions, and what I'll do is I'll pose the question to one of you, but then maybe at the end, if you want to weigh in, the other one wants to weigh in and share some different thoughts. We just want this to be a good free-flowing discussion. So, Dr. Ting, you mentioned in-licensing. Can you say some comments on what the major differences are in conducting research in academia versus industry?
Yeah, so actually, so I'm in Cleveland, Ohio, and Case Western University actually approached us several years ago about trying to develop a master's in biotechnology. And one of the things that they asked us was, what do you think the major differences between academia and industry are? And to me, one of the first things was quality systems. Now, most of us, we were all trained in academic labs, and we know how to do research, but there's a difference when you move into industry. And I think one of the major issues is the quality systems that have to be put into place. For example, when I pick up a pipette in the lab, I'll know the whole history of that pipette. We have full accountability of when it was calibrated, when it was purchased. That's all logged and documented, and I never did that in my academic lab. So it's those little things in terms of trying to maintain a state of control, as my head of quality would say, that are critical, that we can have reproducibility of our cellular product as we move forward. I think that's one of the major things that I emphasized in terms of things that academia doesn't think about, but industry thinks about a lot. The other thing, the major things I notice are raw materials. I think Isabelle's very familiar with GMP manufacturing. A lot of times the materials that academics are using are not GMP products. In fact, they probably never even heard that term. And so again, it's something that they need to be more cognizant of in terms of when they're developing their products. Are there GMP grade reagents? Are those available for their unique product? And again, the other thing is how complicated is it to develop their product? Have they thought about the logistics? How are they going to get it to the patient ultimately? And again, it's early stages. Again, you're just trying to get a concept or just to get your novel product into the clinic, but it helps to start to think about these things earlier. I think the earlier you think about it, the easier your path will be as you're going forward. I'll let Isabelle comment now.
Yes, no, these are extremely good points, and I guess as an academic, a representative of an academic center who has been in the field for almost 2 decades now, we have learned to pay attention to what you just mentioned and the sources, the sourcing of reagents and the quality of the equipment and the control of the equipment. And I agree that you cannot be an academic center thinking about a clinical trial phase 1 in cell therapies and not thinking that and not doing your clinical trial with the expectation that it's not going to move to phase 2 and beyond. So you really have to put some thought into in the developing a process that at least will have where you will have selected your raw materials and your reagents in such a way that the possible industrial partner who is going to come online at a certain time point will not have to change the entire process that may change basically the attributes and the quality of your product. So I can definitely relate to what you've highlighted, and in addition, so what we are providing at Sloan Kettering in my laboratory is a support to our investigators who want to develop cell products and cell therapies early on, so that we can actually advise them on the type of reagents that they can use to produce and manufacture their products, although they don't necessarily think about their research early on as becoming a product, but we definitely help them to select their reagents so that we can support them early on to develop a research product that is going to be compatible to become eventually an early phase and a later phase cellular product. So this is the kind of thought that we have learned to put into the processes so that we can accelerate the development and not create havoc when we're transferring the process to a potential industrial partner.
Very good. Very
Yeah, no, and again, Shawn, I'm coming on top of it, it's also important in terms of when you're thinking ultimately about manufacturing, the scale. Now, for autologous therapies, again, that's a totally different type of production. But if you're thinking about an allogeneic cell product, one has to also consider, is the process you have scalable? Can it meet the demands? For if successful, could it meet the demands for all the patients who would require that cell product? So again, that's something you typically don't even think about in academia. You're just trying to get enough product just to run your phase 1 clinical trial.
Yes, that's a very good point. Absolutely. Scalability is a key component for sure.
And even scalability in the for the autologous products can pose some challenges. For example, in the case of autologous therapies, in most of the clinical trials for CAR T cells, you will have a dose escalation, and you potentially have differences from the start of the dose of 1 log and sometime beyond. And so you have to be cognizant also of what type of bioreactors you want to use for your process, so that this bioreactor can be compatible with the low dose, but also the highest dose of your clinical trial, and so that you do not also introduce additional changes in your product, such as how many days you have to have your product in culture, or cell densities, or any other parameters that may have an influence on the phenotype of your product at the end of the culture. There's enough discrepancies already between the patient sourcing material that you do not want to introduce additional differences that may really muck up the interpretation of the data in terms of clinical outcomes and correlative studies.
Yeah, no, I mean, I think that's very important. And again, for example, Athersys, we started off growing cells in 10-layer cell factories. And that's great for getting to a phase 1, but we're currently in 2 phase 3 clinical trials, and we're dosing at 1.2 billion cells. And again, if we're going to be commercially successful, then we're going to have to, grow a lot of cells. And so, we've taken steps to do commercial manufacturing, moving to bioreactors. We actually have 2 process development teams working full-time, one in Cleveland, Ohio, and one in Belgium. I mean, it's a full effort to really focus on process development, to be able to get commercial manufacturing so that we can support partners. We have partners in Japan and potentially future partners in Europe. So again, you know, we have to think about all these things. And if, you know, if we had thought about it earlier, maybe if we had the bioreactors, like you said, Isabelle, in the very beginning, it might have been easier. But of course, we were in a rush, want to get the clinical trial done. We had a process that worked great in 10 tiers, so of course we were going to use that process.
Yeah, and I'll use that comment to go on a slight tangent to talk about the process development also in the academic setting. For most part, and I think it's not limited to Sloan Kettering, there's very limited funding in academia to do process development. And so it's kind of the egg and the chicken, basically. You have no money to really support an extensive process development in the academic setting, and then you're expected to develop a process that will be compatible with, you know, commercialization down the line with industrial partners. So I think one of the challenges that need to be overcome, and maybe that can be resolved by, in part, getting some early industry academia partnerships where potentially your industrial partner can help you also to develop a process that you can implement and which is reasonably can be reasonably implemented in an academic setting. And we are actually working along these lines with some of our industrial partners, but it becomes also very challenging in the academic setting to have multiple industrial partners who have different processes, and
Yes.
And then you end up with 5 different cell washers and 3 different type of bioreactors. So it's not as easy as identifying the issue. It's also, you know, how much space and how much training you can ask your personnel to absorb, and how many hours you can train people on different processes and for different projects.
I mean, that's again an interesting comment in terms of when do you make the decision to do everything in-house, and when do you make a decision to work with a commercial manufacturing organization or a CDMO, who might, like you said, have some expertise in process development, might be able to help you. Again, it's all expensive, but you know, doing it in-house can be equally as expensive.
Do you guys see any, you know, you've talked about going from phase 1 up to commercialization. Are there key hurdles so you get phase 1 started? What do you begin thinking of next? And how do you plan for moving to and through the phases and into commercialization?
Well, actually, Shawn, I think we should take a step back. I mean, really, you know, how do you even get to phase 1? I think a lot of academics don't necessarily know how to walk through all those steps.
Yeah.
And, you know, there's different regulatory agencies, obviously FDA here in the US, but EMA in Europe, and PMDA in Japan has different regulations. You know, Athersys has worked extensively with all 3. We start off obviously in the US, since we're US based, so we worked with the FDA. And I think one of the nice things is CBER has been very easy to work with. They've issued a lot of guidances for the space. In comparison to the drug world, the small molecule world, there's multiple ways you can have interactions with the FDA. For example, they have this thing called Interact, which is sort of like a pre-IND meeting where you can bring up a question. For example, you know, Isabelle, you could say, is this process, is it okay to make a product that I can put into patients? Then they have a pre-IND meeting where, again, the ability to ask questions. Is this the appropriate animal model that would enable an IND? So these are things, again, that the academics typically haven't thought about, but they do require time and effort. And I think the main thing is when you're dealing with regulatory authorities, their main focus is really, it's about patient safety and well-being. And so have you really done all the studies that satisfy those requirements? It takes a lot of effort, it takes time, but it's important that you get it right. You don't want to keep going backwards. You want to make sure you listen to the regulatory authorities that are there to work with you, and you do it right. You put in the effort. And I think that's kind of something the academic world needs to think about in terms of moving forward before we even get into the phase 1 space. I don't know, Isabelle, do you have any thoughts on that?
Yeah, no, definitely, and also maybe because I've been thinking about these issues for the past 15 to 20 years, and we have performed many different T-cell trials and CAR T-cell trials at Sloan Kettering. We have learned also to use these resources and to get these interactions with CBER and with the FDA, and to ask questions during pre-IND and now interact meetings. And I agree, it's really important to take advantage of these interactions because the agency is very much willing to give you feedback on your proposed plans and your proposed manufacturing plan, as well as quality plan, and also the farm tox studies that you can perform on your products. And I think the pre-IND is very useful and prior for an academic institution or an industry, an industrial partner to perform these studies, and prior to performing these studies to get the feedback from the agency as to if your farm tox plan is adequate, and is if the cell doses that you're planning to use, the time that you're planning to monitor your animals are adequate and satisfy the agency requirements.
So, sorry, Shawn, what was your question again?
I was, well, thank you very much for taking the step back and kind of walking through all of the different things. My question was kind of going along on the theme of what do you begin planning? How do you plan your steps going from phase 1 to phase 2 to phase 3 from your, when you take something from academia, say you have a phase 1 academic study coming in and you want to partner it at Athersys and bring it into phase 2, or from Memorial Sloan Kettering when you find an industrial partner and you want to go to phase 2. Can you guys comment on that?
Yeah, I can start if you want, and maybe Tony can take it a little bit further, because I will stop much before the commercialization process. So in our case, I guess at Sloan Kettering, we're mostly focused on phase 1 clinical trials, and maybe phase 1, 2. I think the reasons for that is first, once you have done your proof of concept in a phase 1 clinical trial, you will likely need to do a multi-center clinical trial. And I think we have performed a couple of those multi-center trials at very limited scale. And it has some qualities which are slightly different from doing an internal phase 1 clinical trials. First, generally clinical centers are quite reluctant to hold an IND and have patients enroll at other clinical centers for reasons of liability and having and in terms of the oversight of the clinical trials at other centers. So already at that stage, it's more desirable to have an industrial partner who will actually conduct the phase 2 trial, which means that basically by the time you're initiating a phase 2 multi-center trial, you will probably have transferred the IND from the academic center to the industrial partner. So I think that's essentially the way we think about it at Sloan Kettering. The second reason to not necessarily want to do a phase 2 clinical trial in an academic center or a hospital is the amount of resource that you need to conduct this phase 2 clinical trial. And so it's not necessarily the mission of an academic center to conduct these phase 2 trials and beyond, except maybe in the case of rare diseases where the number of patients may be very low, and where potentially you would want to get products commercialized for a very specific indication with very limited number of patients.
No, definitely valid points. I mean, going from phase 1 to 2 and 3, obviously, again, scale, do you have enough product? Can you make the product? We work with CROs who help us do a lot of the clinical trial management, because if you're in multiple sites, someone to basically do all the monitoring, manage the clinical database, the data management, the pharmacovigilance. It is expensive, and it is something where you have to really keep track of a lot of moving parts. Just going back to the manufacturing, when you license something, you have to really think about what's the vision. How do you want to build it yourself? Is that really the way to go, or do you want to just work with contract manufacturing operations? And the regulatory roadmap. So one of the interesting things is working again with the FDA, for example, that there's a lot of different steps one can take along the regulatory pathway. For example, you can get fast track designation, which allows you to have more conversations with the FDA. And more recently, because of the 21st Century Cures Act, there's something called RMAT designation. So that's what's it called, a regenerative medicine advanced therapy designation. And so again, that gives you a recognition that you're working on a cellular therapy, that you have demonstrated some benefit, and that it's in an area of unmet medical need. So recently, actually it was yesterday, I think, Athersys, we announced that we had gotten RMAT designation for our multi-step product for the treatment of ARDS, or acute respiratory distress syndrome. And currently we're running a phase 3 COVID-19 induced ARDS trial. So the nice thing about the RMAT is it just gives me the ability to have more conversations with the FDA about regulatory strategy. And one of the things we may think about is something known as an SPA, or special protocol assessment. So this is a very interesting thing. So we are currently running a phase 3 stroke trial under an SPA. And so what that allows us to do is just one phase 3 clinical trial to potentially get market approval, as opposed to traditionally with the FDA, you have to run 2 phase 3 clinical trials. So again, the regulatory environment is something you have to think about in terms of your strategy as you're advancing things from phase 1, 2, or 3. So the FDA has been very easy to work with. Japan actually implemented a whole new regulatory system just for regenerative medicine products, and this actually allows conditional approval. And so we have a partner in Japan who's currently conducting trials for both stroke and ARDS, and so they may get approval before we do, but it would be a conditional approval. So that there is some differences, again, depending which countries you're working with.
Thank you very much. That's very interesting. So I'm going to switch a little bit. We talked about kind of what you think about when you are transitioning one back and forth. What are some of the possible models of partnerships between academic centers and industrial partners? Are there things that you've seen that have been successful, things that you've seen where you've had lessons learned? Can you comment on that? Maybe start with Dr. Riviere.
Yes, sure. So I guess one of the models that we have been able to work with is you have a new molecule such as a chimeric antigen receptor, which has some give interesting properties to a T cell and makes that T cells more potent in terms of anti-tumor efficacy with a long-lived profile. So you can, if you license this technology right after you have done a proof of concept in an animal model, you can basically get support to do additional research, but also process development and preclinical research with the support of your industrial partner. And this allows you to kind of have an interactions where if your partner is already involved in cell therapies, you can get some of the feedback and some of their expertise into developing a process for this particular cell therapy, and therefore you can have likely a better process and something which is will be ready sooner to enter the phase 2 and the pipeline of commercialization from the point of view of the ability to be manufactured according to the industry standards and not just the academic standards. So I think that's a model where if you can license your IP early on, you can definitely have a longer partnerships and an earlier partnership that will help improve the probably the product development. Obviously, this is not always possible, and sometimes also if you know you have something really hot and you have the resources in the academic center, you want to give some more value to your products and you want to develop it a little further and get some phase 1 results that will be, if successful, will give some additional value to your IP, and we have done that with other industrial partners, and there you have developed a process, and you can tweak your process with your manufacturing process with your partner at a later time point. It can be, you know, maybe it's good, maybe it requires some additional work. And if your partner is going to do a phase 2 derived from your phase 1, they may have, if they have to tweak the process, they may have to do some comparability studies and redo a phase 1 if the process is really difficult to transfer, or if it's only some mild or small changes or dim small. Maybe you will be able to, the company will be able to launch a phase 2 and based on the phase 1 data collected by the academic institution. So I think these are 2 models that I can see would be that we have used and which I think have some values. Another one which we have not really experienced would be if a company wants to run a clinical trial at a center and has already a process that they want to transfer to the academic center. In that case, the process is transferred from the biotech or the industrial partner, and that's using the academic center more as a CMO. I think this is probably feasible as well.
So, you know, Athersys, we have our own technology. We have our own IP, and I'll probably come back to IP at some point. In terms of for us, in terms of partnering, and we do have, or historically we have had partners in Europe, and we do have a partner in Japan. And so it's very interesting in terms of, you know, we basically license out our cells so they can use them. And some partners are very interactive, and they want to, you know, work with you, and they're hand in hand because they know we're kind of tied at the hip. You know, our cells is their cells. Some partners may just want to say, treat it like a small molecule. They've been licensed, it's theirs, they do whatever they want with it. So it does vary in terms of the dynamic that one has with the partners. And I think in the case of cell therapy, because it's still fairly novel, and there's a lot of biology that we don't fully understand, I think it's important that whoever licenses or partners, that you maintain the relationship with the academics who did the original biology, because there's a lot of science that we're still trying to understand about these cells and how they work.
Yeah, definitely. I mean, if you do this type of partnerships between academic centers and biotechs or pharmaceutical companies or industry partners, there can be an evolution of these products over time if you keep some ties, so that you can develop, you can have a pipeline that will improve over time, if that is what you can do or wish to do, and this can definitely. I mean, I think nowadays a lot of pharmaceutical companies in particular have decreased their early preclinical research, and they really rely on the partnerships with academic centers for the innovations in terms of research. So I think there's a tendency to get IP and licenses early on in the pipeline, and take advantage for the pharmaceutical industry to take advantage of the innovation in academic centers. And I think because of the limited resources in the States at the moment, and probably for some period of time in terms of research funding, it really also becomes an important resource, or important resources for academic centers to be able to conduct their research. So of course this comes with also strings attached, but these are very, can be very productive and important partnerships for both.
No, I agree. And, you bring up an interesting point in terms of IP. And again, it's gotten a lot better, but I think, in the academic world, they're not only as nuanced about the capturing the intellectual property, as well as really surveying the intellectual property landscape to make sure that they actually do have something novel. Because, if you eventually want to partner with an industry or to raise money, even, it's an imperative that you have the IP captured appropriately. I mean, that's something that we've learned very early on, to make sure we capture all the IP that we can possibly capture.
Yes, absolutely. And I mean, sometimes academic centers do not have the resources to do the survey of the landscape. At least that's what we're told when we want to file our IP with the institutions. Or there's not always the resources to be able to do all the due diligence and survey that we would like to do.
Can you guys each speak to kind of high level the process? Because I know when you're going from academic centers to industry, the universities typically have a technology transfer office or something like that. And then there's the licensing that needs to be done, not only with the academic lab, but also with the university itself. Do you have any advice, tips, tricks, things to avoid, things that went well?
Yeah, I mean, I'm obviously not directly involved into these aspects, but generally, yes, there is a discussion in terms of IP agreements between the academic institution and the industrial partner. And generally, the institution is very good at shielding us from these discussions, and I think it's probably for the best of the academic integrity. So generally, we leave it to the lawyers and to the tech transfer offices to discuss these agreements. What we do is we definitely then develop very detailed statement of works that will highlight what the milestones and the timelines and the deliverables will be over time if we are obviously supported by an outside entity, and this is very useful for us to be able to structure the, I mean, this is a very different way of structuring the research. I mean, when you are an academic researcher, there are different ways you can do research, and for the longest time I was doing research without necessarily the goal of developing therapies, but more about the learning about the biology. But if you are going to develop a therapy with an industrial partner, your milestones are going to be very well set, and there will be beautiful Gantt charts that will hold you accountable and productive. So I think we have learned to really work out these agreements which make us live and breathe, on a day-to-day, and we understand the usefulness and of project management.
Yeah, no, I mean, I think, like you said, it's usually the tech transfer office that you're dealing with. The inventors have already, made their agreements with the university typically. And so it's all about, working with the tech transfer office, and then, like you said, coming up with like a material transfer agreement, a scope of work, the milestones. These are all fairly standard.
Very good. So I guess another question that I'd like to pose to both of you is, we've talked about the different aspects, thinking about a particular case study, and you don't have to go into the formality of a case study, but think of a technology transfer process, and can you highlight what went smoothly and what was difficult, and then how you would change? More specifically, going from academia to industry.
So, I mean, I can speak hypothetically. So when we receive an academic product, like I've already talked about, the quality systems typically weren't in place. There might be a publication if you're lucky, but a publication describing the development of the technology, or how to grow those cells, or it doesn't necessarily capture all the little tricks that you learn in the lab. So, there might be an incubation where you have to do it for 5 minutes, but they don't mention you have to swirl for 5 minutes. So it's those things that when you do the tech transfer and you don't really capture all the nuances, and you try to repeat things and they don't necessarily repeat right away, you have to go back and have that discussion. And there's a lot of back and forth. Okay, we did this, it didn't work. Oh, well, did you do this? It's like, oh no, it wasn't written down. And so it's that's where I mentioned the quality system, is that they haven't truly captured everything that's involved in the process. And I think to make things smoother, it's almost beholden to like sort of sit down and watch somebody go through the entire process in their lab, and so you can make it into an SOP. I mean, Isabelle, do you have similar experiences?
Yeah, definitely, and I think that I would say there is successive steps which go, more and more in depth into the process. And if I take my, collective experience, you can basically we will get a protocol which may be already an SOP from another institution or an industrial partner, and we will definitely have both the manufacturing team as well as the process development team. I mean, for us, it's the process development team is also the manufacturing team. They will read the SOP in depth. We will also have our quality assurance group who will do the same, as well as the quality control. They will review the sampling plan, and they will look at the testing required. And generally, we will kind of readapt the process to our quality assurance program, as well as and tailor it to our quality control. So we may want to add certain tests, or we may want to remove certain that we're not able to perform at the center, or that would not be coming in a timely fashion. So there is all this discussion at the very detailed level, and then we will basically rewrite the document according to our internal quality system. And if we get a process earlier on, we will definitely, as you stated, ask a lot of questions and introduce additional details that were not captured in a research protocol. Sometimes you'll be surprised that, yes, you have to go all the way to observing the person who is doing the process to really capture what it is that was missing and that somebody was taking for granted. Because if it's not written, yeah, it's not going to necessarily be something that is going to be intuitive for everybody. There is a certain level of art in growing cells, and you want to remove the art part.
Yes,
from
exactly.
the documents.
Yeah, it should not be an art, it should be a procedure.
Exactly.
Yes, very good. So I'm going to shift a little bit over toward regulatory strategy. And so I have a two-part question. One, Dr. Ting, you talked about this early, about the FDA interactions and stuff. Can you speak to when you would recommend getting an interact meeting in your stage of development? And as you go along these, if you're thinking from a researcher, I have a new product, I've developed this great cellular therapy, when do I begin approaching the FDA?
Yeah, no, I mean, I think it's important to figure out what the question you really want to ask. So for example, let's say you started in a novel technology and you're doing viral transformation of a T cell. And you want to know, is my production of the virus, is that of the quality that whatever I do to these cells, and can I put those in patients? So that may be a very simple question. You could just go and have your interact meeting, and they could say, well, no, this process is not, of the standards that are necessary to put these cells, which have been virally transformed, into people, and you need to do, steps 1, 2, and 3. So I think those are important to have with the interact. The pre-IND, the way I think about the pre-IND, again, it's about the manufacturing process, how appropriate it is, and also in terms of the efficacy studies, do you have the quote GLP IND enabling animal study performed such that the FDA agrees? Yes. If you see safety, and efficacy in this animal model, we agree that can support an IND. And I think, we've had very good interactions with the FDA where they'll say, this model's okay, but maybe you could do a few more tweaks to it. And it's good before I spend, $300,000 on a study that I know it's going to, supply the data that supports an IND filing.
Yeah, I would echo what Tony said. I think that for checking on your manufacturing, if you have a new process that you're implementing, if you're, for us, we went from viral transduction to gene editing. And so what is it, if you do a gene editing, what are the steps that or the quality control or the testing that you would want to do as part of your farm talks or as part of the pre-clinical. You don't want to have to do so much quality control, for example, on each product, but then you need to ensure that the reagents that you're using for performing your gene editing are of the quality that will allow you to not do all these quality control for every product. So you may want to do it on your validation products, and you may want to do an in-depth analysis of your validation products for various off-targets or cytokines growth or deep sequencing or whatever would deem to be necessary to ensure that the products that they're going to generate for the patients are going to be safe. So I think it's really in terms of safety and efficacy. And I think, as I mentioned earlier, yes, as you stated, the farm tox study is really expensive. So you want to make sure that the agency is going to be on board with the design of your farm tox and that they will endorse the timelines and the cell doses that you have proposed, as well as the additional testing that you're proposing.
Very good. So when you're thinking about partnering with an industrial partner or transitioning from academia to industry, what would each of you say are the key elements that you want to make sure an academic IND contains to make a smooth transition? You've touched on some of the elements such as the farm talks, the GLP studies, et cetera. Can you expound upon that topic?
So I think that what I can speak to is to, first I think you need to have an alignment of the culture also of how you want to work. If you're going to co-develop a product with an industrial partner, I think, and you're going to have these weekly meetings and progress on the research as well as the process development and eventually developing a product that will be infused in subjects. I think you want to share some of the not only the vision in the science, but also the goals of the safety and the efficacy, and you want to really be aligned on these parameters. We have, I think that in my view, the success relies both on the technology that you're licensing, but also on how you think about what are the best parameters to measure the efficacy and safety of these products.
No, I think you touched on a point that I think is also very important, is the culture, so that there's a good fit in terms of the industry and the academia. Do they have the same vision? Do they have the same goal for how they eventually see this product going into patients? And so I think that's an important step that one needs to have, that level of trust as you're co-developing, like you said, Isabelle, product to market. In terms of so I typically don't look at academic INDs, so I actually don't know what goes in them, but but I obviously it would be the things that you had already mentioned, Shawn, is the farm tox section, the CMC, basically is the product safe, and is it well controlled?
Yeah, and I think we also touched earlier on in the CMC section on the sourcing of the materials, and and how materials have been, raw materials are looked at. I'm lucky to have in my team some quality assurance managers, and who are very experienced, and you can't you can't take for granted what is sometimes written on a CRV. If it's again, it's the art of writing certificate of analysis can be sometimes can be surprising, and you cannot just assume that if it's been tested, it means that it was negative. And so there is definitely a learning curve also for academic centers to understand what type of raw materials they're working with. Or a company who claims that they're making GMP products, but you know, what were the qualification? What were their own qualifications to call
No,
them
yeah. What is GMP exactly?
What is GMP? GMP can be interpreted in different ways, and we want to make sure that you're aligned with what it should be in terms of quality and control of and the results that are proposed on the certificate of analysis, and or you know, for that matter, how the process is conducted in the plant. And in academic centers, we don't typically physically audit the suppliers, but I've learned over time also that it can become necessary to be really involved in, and to really not only just rely on a certificate of analysis sort of compliance, and to go deeper, because you can have some disappointing surprises.
Agreed.
Very good. So we have just over 10 minutes left. Would each of you like to give a take-home thoughts, or we can continue having discussions, just free-flowing?
I think the free flowing's been going pretty well. I mean,
Okay.
I think you've covered pretty much all the topics. I don't know. Do you have any other topics that we haven't covered, Isabelle?
That's one of the questions. I'm looking around here because I think we have covered most. Let's see. This is a good one. What would you consider the best way to handle, and this talks about culture and also philosophy, if there's a disagreement on an interpretation of the guidance? What advice do you have on that? You know, some people say, oh, well, that's just way too advanced for this stage of development. Other people say, no, this is a game changer and you have to have your impurities profile.
So if I understood the question well, is a disagreement between the industrial and the academic partners, or?
Yes, yes.
I mean, I think that, you know, life is a compromise from from the day we're born. So I think that you have to stick to what you think is the best practice from where you're standing. I think that there are different interpretations of the GMP. If you are working in academic center versus industry, I mean, we're not, academic centers are not held to exactly the same standards, because we're not commercializing, we're not performing a clinical trial in the early stages with the intent of commercializing a product. So the incentives are different, and in addition, the regulatory agencies note that we do not necessarily have all the same resources that industry has in terms of how to perform studies. I mean, sometimes we can get away with doing a GLP toxicology study, but we can do a non-GLP pharmacology study, for example. This can be tolerated as long as we can document, you know, how the experiments are done by way of SOPs or protocol, and you don't have to do everything under GLP, or I mean under GMP. if you can also provide documentation and further characterization of certain raw materials, for example, or so I think that you know, the key to resolving some type of differences or disparities between academia and industry have to do with providing the documentation and the explanations which will demonstrate that your specifications or qualifications are reasonable based on the safety, efficacy, and level of resources.
Yeah, I mean, I think, again, for phase 1 clinical trials, you know, you probably don't need everything to be a GMP raw material, particularly if studies done in the academic setting. Obviously, if there's conflict between the academic and industry, I would support the industry partner. But I mean, when you look at some of the guidances, there is some wiggle room as to you know, ways to go. I would say the industry is typically more risk adverse. And so we go the route to avoid risk is probably the challenge there. While the academic may say, you know, maybe we can take that risk. So it's really the balance, like you said, how strongly do you feel about it? But I would say from the industry perspective, it's usually going to always be more risk adverse.
Very good. So looking at the future, it's, you know, we all know how well our predictions for 2020 have gone. So pull out your crystal balls and predict about the future of this field.
I mean, as I already said, I'm really excited. I think, you know, Isabelle's in a great space. The immuno-oncology is just, it's growing tremendously. And I think, you know, it started off with the CAR T or T cells, but now you're seeing all sorts of immune cells being evaluated, you know, NK cells, macrophages. It's quite exciting, I think, to be in this immuno-oncology space at the moment, and it's really, you know, to be honest, it's great because cell therapy has been around for several decades, but it's, you know, I think a lot of the big pharma never really appreciated it, or again, it's complex. These are cells. It's not a small molecule. It's not a protein. It's not an antibody, and so, but you cannot deny some of the therapeutic benefits that have been observed in the clinic. And I think that's finally gotten the attention of the larger pharmaceutical companies. So I do think because of that, the whole field has an energy and excitement to it, that it's really a great time for cell therapy.
Yeah, I can only echo this. I think these are very exciting times. I think that we have made the proof of concept that these cell therapies and genetically modified cells can be efficient. And I think we now have to demonstrate that they can, well, they've been demonstrated to be efficient mostly in the hematological tumors at this point. I think we definitely need to have some breakthroughs in solid tumors to demonstrate the more broad applicability of the platform. And I think that the next step is to definitely reduce the cost of the therapies. And one of the ways to do that is to develop some allogenic platforms that will allow the treatment of multiple patients with one product. I'm cautious not to use the universal product. I think that even if we needed only 50 or 100 products to be able to treat most of the patients for particular indications, we would already have made large progress. And and reducing, yes, the time that it takes to make these products, and foremost also ensuring their safety.
I think we're nearing the end. Do either of you have any like technology breakthrough wishes to make your field better? Anything you want people to come up with? Any last comments you want to make?
Sure. If somebody out there can preserve cells without cryopreservation, I will definitely be, the beta test or the alpha test, or even testing the prototype. I think this is one big hindrance in the system right now, because it creates all the cold chain challenges, and so yes, I can leave it at that for now. This is one of my
That's your dream, to have a room temperature cell product.
What about you, Tony?
I think, again, I think the advantages of the genetic engineering, I think we're going to have really highly specific targeted cells that we can develop for, various therapeutics. Again, it's a lot of biology, but I think it's there. And I think the development of really specific targeted cells is going to, personalized medicine is the word, but it's really going to make huge advances for the field.
All right, very good. Well, I think that brings us to the end of our time. So thank you both very much. I have learned tremendous amounts, and this is very helpful. So thank you both for taking the time to participate in this session.
Oh, thank you. No, it's nice to meet you, Isabelle.
Nice to meet you, Tony, and thank you, Shawn.